Listen on iConversation, man.
Charlie Rignaia.
So, hello, Jonathan.
Hi, Charlie.
Welcome to the show. I very much appreciate you being here. I know you have a very busy schedule, so I much appreciate that. But let's start with who are you and what you've been working on for the last couple of years with regards to ALS, Parkinson's, and Alzheimer's disease.
Yeah, thank you. I mean, thanks for inviting me. It's nice to come and talk to your audience. Well, as you can hear, I'm an English man, born in '69 in a place called Chesterfield in the middle of the UK. Pretty much, yeah. People always say, what's the closest football team or which football team you support? And I say, I don't like football. I don't really follow football, but it would be, yeah, Sheffield United again. Oh, Sheffield's the nearest big city. Manchester's about 60 kilometers to the west.
You're a weird guy. You're from England and you don't follow any football.
I used to be. I was a mad Liverpool fan, in the '70s when they just won everything. Yeah. And I kind of got a bit bored. I mean, I had everything: Liverpool tracksuit, mug, everything was Liverpool, and I was crazy about them. But then partly they won everything, and they also. I moved schools when at 11. I went to a different school. I went from this very protected, nice environment to this hellhole with like 1,500 students. And if you weren't a Derby fan, you were literally hunted. So I had to switch my allegiance to Derby County, and they had been good a little bit in the '70s, but nah. So, and at that stage, I started to play rugby, and I kind of, I came from a rugby family, so rugby was like the game that my dad played and my brother played, and I loved rugby. I mean, I was school captain at the age of 11, 12, 13, but then my testosterone came way later than everyone else's. So I was like this little stringy leg boy and trying to tackle someone that was like 90 kilos running at full speed down a pitch. And I just got flattened. So I kind of dropped out of rugby as well. Probably directed me more into science, maybe. I don't know. Not sure.
It could have been the spark.
Yeah. Anyway, I come from Derbyshire. Which is a very beautiful area of the UK. It's the Peak District. It's the second most visited national park in the world after Mount Fuji, because it's very close to almost 60% of the UK population within maybe 2 or 3 hours. But in the week when nobody comes to visit, it's just spectacular, beautiful area, lots of lovely walks, great pubs, nice things to see. So if you're ever in the UK, I recommend going to visit Derbyshire. Then I was very interested in science from an early age. I loved it. I always said to my mum, I'm interested of nature. And I think I was, on a track to be a scientist from childhood, really. And then I went to university in Leeds to study microbiology, virology. And then after that, I traveled around the world for almost 2 years. I took a kind of gap period. And then when I came back, I couldn't think of anything else but the research project I'd done during my degree studies, which was trying to clone a gene that was coding for the outside protein, like spike protein, we're going to talk about later, but from herpes viruses, from a herpes virus that infects horses and causes a lot of problems for kind of abortion in mares, which are kind of racing horse breeding mares, for example. So, there's a lot of economic interest in studying this virus because it's cost people money. So for me, being at the bench, doing experiments, designing experiments, seeing that they work or didn't work, and using your brain to then try and work out how to do it better or find out what you wanted to know was just almost addictive for me. So I found that really stimulating. And so when I came back from my traveling years in Australia and Southeast Asia, I decided, I want to do a PhD. It's the only thing I can think about, and I don't want to wear a suit. So, that precluded me going to work in the city in finance or in a bank or something. So I found a PhD in a place called the National Institute for Medical Research, which was in northern London, a place called Mill Hill. And this was like the flagship research center for the MRC, Medical Research Council, which is like Swedish Vetenskapsrådet, VR, although a lot better funded probably than VR, at least at that time. It was the sister institute to the LMB in Cambridge, which is where Watson and Crick, made this great discovery of the structure of DNA, which was, set the scene for molecular biology for the next period from the 60s onwards. 50s, but really the 60s when it took off. So I went to Mill Hill to do my PhD, and I wanted to do something which I thought wasn't going to, die out in the next 5-10 years. And at that time, developmental biology or embryology was just exploding. So the study of how you go from a single cell embryo, a fertilized egg, to an adult or a fetal baby body form. How do you go from one cell to something that's got legs, arms, eyes, ears, a nervous system? Everything's in the right place usually, and everything's connected up and functioning as this beautiful, creation. So I just found that a fascinating subject. So that's what I went to study in my PhD, which was really how do cells in the body know where they are, so that they know what they're going to become. And we were studying this real level of the body in mouse embryos. How does a cell that's going to contribute to the shoulder, let's say, and then also to the nerve cells in the spinal cord here that innervate the shoulder, and the muscles that are around this region to support it. How do those cells that make those tissues know where they're going to be? Why aren't they further up or further down in the body? And then I worked with mice. So I did a lot of, cutting and pasting small pieces of DNA into little plasmids so we could grow them, and then study injecting those into mouse embryos. And these pieces of DNA were carried a kind of molecule that they could turn on and off, which gave a color, so we could see where this bit of DNA was active in the body plan of the mouse embryo. And we were trying to understand, how does this know it's in this place and not further forward or back? So that was, a very intellectual, a lot of technical things, using your mind to kind of work out how to make these constructs that you were going to test, and then also the kind of dexterity skill that you needed to inject these into a tiny, tiny mouse egg, or into the nucleus of a tiny mouse egg under a microscope, and then put those eggs back into a mouse and let them go develop to embryos or be born, and then study where this gene was turned on and off in those offspring. Thing was, I got allergic to mice in the process. And
so you didn't come to a conclusion before did you have to end before you came to a conclusion?
No, I got to the end, but it was a big hindrance to the last year. I mean, in the UK at that time, PhD was 3 years, and that's all you got basically. So I had to, a lot of the work I was doing at that time, I wasn't able to finish off, or at least I couldn't continue after my PhD in that field. I had to change. So then I went to work on, but I got very interested in how the brain develops as a part of that. I didn't really know what the brain was at that stage, what it came, which tissues it came from, how it formed and stuff. So, many people don't, unless you spend your life studying that. Why would you? But I got interested in the brain and how it formed. So I went on in my postdoc to study brain development. And this time I switched model system to chick embryos, because then you can work in an egg. You don't need to sacrifice, mice. It's not that I don't really like, experimenting on animals. There isn't really an alternative for some fields, but these mice living in a mouse house, and they're bred for that purpose. But I didn't really like the idea of that, so it felt better working on eggs, because it's less. It's still an animal. It's just less cuddly. But then I got through that work. I got allergic to eggs.
Oh, okay. That's a bummer.
Yeah, major bummer. So late on in that work, I couldn't really continue either.
Can I ask you a question? Do you think you got allergic because you were exposed to it a lot, or?
Yeah, I do. And now thinking back and knowing what I do after the last 5 years, and the role of vaccines and immunity and autoimmunity, if I think back, the two occasions when I've developed, so I was allergic as a child. I had kind of cat allergies and things. So my allergies have developed. I become allergic to things that I work with intensely. But those two occasions when I got allergic to mice and then to chick eggs was exactly within a few months after getting vaccinated. I got vaccinated to go to Africa. I went traveling there when I was doing my PhD with several, yellow fever, a bunch of 4 or 5 different vaccines. When I came back, that's when my mouse allergy started to develop.
Mm.
And the next time I was vaccinated for hepatitis B because I was working with human tissue as well as chick embryos. And that's the time I got allergic to eggs. And I never connected the two together really until more recently. I'm not saying that that's definitely the cause, but I don't get allergic usually to other things. Mildly, but not so that was severe, severe anaphylaxis. Basically, I was extremely allergic to eggs for a while.
Yeah, that's one of the things RFK Junior will be investigating, right? Allergies connection
Yeah.
to different types of vaccines.
Yeah, and I think it's so necessary, Charlie, because I bought into the. Oh, before COVID. I'm fully vaccinated. Everything came along I needed. I took it. The kids are, the family is. There's no anti-vax sentiment here at all. But I realize that all the studies that have been done now, there's never been controlled against. All the vaccines on the market have never been run against a placebo control, ever. They're,
Yeah,
they're benchmarked against the last vaccine.
yeah. I've read his book. I've read all of his books. The Real Anthony Fauci, The One Cover Up, and the Vax versus Unvax. And it's crazy how they haven't done a placebo-controlled double-blind study against an actual placebo.
Yeah.
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 But go on. Sorry.
Yeah, no. So anyway, my career working on chick brain development kind of started to hit the buffers. But actually, around that time, that's when I first met my wife, or my now wife. So I went over to a conference in Colorado in the US in Keystone. So one of the perks of research, there aren't many, but one of the few is that sometimes you could go to conferences in exotic places. And we used to go skiing quite a lot. So we'd find conferences in ski resorts so that we could go, spend a week in a conference and also ski. And the Keystone meetings were very well known for, I mean, beautiful area as well, very, very high quality science. But, in the daytime, you got to ski, and then the conference was in the night between like 3 p.m. at 10 at night. Pretty tough if you fly in from sea level in London up to like 3,000 meters, and then you ski all day, and then you've got to stay up all night to listen to lectures and things. But well worth it. Anyway, that's where I met my wife. She's Swedish, and I moved over there for a year to be with her because she was doing a postdoc in Stanford University in the US. And then we came back to London for 3-4 years, and then it's got to a point where I couldn't really carry on, or neither of us could carry on being postdocs anymore. We needed to either leave science or find a kind of group leader position, or a leadership role. And we both got positions in Sweden, so we moved here 2008 and started our own groups. Eventually, my wife gave up science; she went to be a teacher, but I carried on, and made my way here up in the north of Sweden as a scientist, as a research scientist, and then as a teacher, a lecturer on the medical program and also on the dental program. I just say as a disclaimer, everything I say today is my own personal opinion, so nothing to do with my employer. Yeah, so I in my day job is scientific research and teaching as well, which is very interesting in this day and age. When I'm very much for the truth. I don't want to hide the truth or not discuss the truth, whatever that truth may be. But it is very difficult because people are entirely brainwashed, unfortunately, especially in Sweden. Not just Sweden, most of the Western world. I think we've been propagandized and brainwashed about many things, not just vaccines. And that's quite distressing, I find, because when you've spent your life really trying to follow a career which isn't very well paid, it's hard work, it's long hours, it's not that rewarding, but you do it because you think you're making a difference, you're contributing to new knowledge and things that might lead to a cure for a disease. And then COVID comes along, and you get the biggest deployment of a experimental therapy ever on humanity, which is clearly created, catastrophe, an enormous amount of damage. and you're not allowed to speak about it, because if you do, you're an anti-vaxxer. It's quite distressing, actually, because you just feel, what am I doing? What have I spent my life doing, and why am I doing this now? Especially because at the time, I've been working a lot on ALS, because it's a disease that really motivated me to try to find a cure, or at least understand what causes ALS. So I hopped a little bit, and because I'd got allergic to mice and then to chicks in London before I moved over to Stanford, I started to work on human cells as a kind of model for human diseases. So how can we use cells that you just grow in a dish, in vitro, it's called, or in a petri dish, in order to try and model things that happen in humans when they have disease, to better understand what is going on, because it's very difficult to actually work on people, right? You need ethical permission. Everyone's different. Any sample you might take will change throughout the day, and it's really expensive. And you need to be a clinician to do it, usually, because you need ethical approval to do the experiments. So I started to work in Umeå, firstly on a different project, which we're working heavily on now, which wasn't so much related to ALS. And then I started to work with the ALS group in Umeå, which was quite a large group at the time. And what we were doing really was, I had a lot of knowledge for working with cells, and also how cells would then develop from early stem cells in the nervous system to more mature neurons, like of the type that die in ALS. They're called motor neurons. So I decided that we could take these. The clinician there had collected a lot of skin samples from ALS patients and frozen the cells down, but didn't really know what to do with them. But what we did, because around that time there was a new method came called induced pluripotent stem cells, or reprogramming, which means essentially that you can take an adult cell from a person or an animal, put in the proteins that are there when it's very early in the embryo that tell it to be a stem cell, and turn it back into a stem cell. So you basically take the cell and time shift it back to the very early stage of development, when it had a choice of making any cell type in the body. These are called pluripotent stem cells, so they can make any cells that you will have in the adult. So we could turn these cells in the dish back to this state, and from those we could then take them forward in time again, or through their developmental program, to make the cells that are deficient or defective in ALS, and then study them in a dish instead of in a person.
And what's a dish, if I can ask you?
Well, you cells in the lab, you grow them in a little plastic bottomed, or a plastic dish called.
Okay, yeah, it's one of those. Okay, yeah.
And then you feed them by giving them some media which contains all the things that they would normally get from the blood supply, and then they get their oxygen from the environment, from the air, and then you keep them in an incubator at 37 degrees, and they'll divide and do their thing. And then if you treat them with different chemical or proteins or chemical signals that they'd normally see through their developmental program, to turn them first into nervous system tissue, and then into the part of the nervous system where they would normally develop from. And then you can start to give them things to say, okay, now you're an adult, now you need to grow up. And then they'll turn into neurons, and they'll grow out long processes called axons, which neurons have. And you can check them with various marker proteins that they they should turn on, to check they're the right thing. You can look at their ability to fire off little electrical signals when you stimulate them, for example. So what we were doing, and towards the end of that project, was to really, what I wanted to understand. So for ALS, motor neuron disease, and also for other neurodegenerative diseases, you have about 10% of cases where there is a genetic mutation in the family, and you can connect the disease to that mutation. But the other 90% are called sporadic forms. You have no idea what caused the disease, usually. So if you want to make a therapy for these diseases, one way is to go in very early, if you know, and then correct this genetic mutation, if you can do that in humans. But there are ethical things around that. Should you really treat someone that's not actually sick yet? Because not everyone that has the mutation will develop the disease. But the other 90%, you don't know what the trigger was. Was it a group of mutations that are not really clear that kind of contributed, so multiple genes, or was it also environmental cues? Something that in the environment, something you ate, something you were infected with, some pesticide, some chemical you're exposed to, or a vaccine. And that's a big issue if you don't actually know what starts the disease process in the beginning. So how can you devise a therapy? We spend millions a year on new therapies, for trying to make new therapies for, say, dementia or for ALS, but they never succeed because we don't really know what the basic problem is. So my interest was trying to get back to the start, to try and understand what is it that triggers ALS in the first place. We know from some forms of the disease that there are certain proteins that clump together and they form these very sticky clumps that are called prions, or prion-like things. And what that means is when the protein forms that particular shape, it can get its friends to do the same thing. And it's a bit like Lego blocks sticking together. And then they can break into smaller parts and spread into another cell and cause that same process to happen in another cell. So it became kind of clear in ALS that this was happening, and we wanted to understand why. What was it that made this process start in the first place? And what we found was quite interesting, actually. We found that low oxygen, so if you took the cells that you'd made through this special process I described and you put them under low oxygen overnight, then they made a much more of this unfolded sticky protein clumps than if you just left them at the normal oxygen concentration that they were living in. And this was kind of interesting because in ALS, there are a few risk factors. I mean, the main one is age. So the older you are, the more likely you are to get the disease. But that's the same for all neurodegenerative diseases. So something to do with the aging brain, makes it more likely to stop working properly. That's not surprising. But the other risk factors are things like smoking, trauma to the spine, stroke, some people that have a kind of malformed blood vessels in the spinal cord or brain, and all these conditions would have low oxygen as for a while at least in the spinal cord as a condition. So we believe, at least using this very simple model in dishes, but we could have found one of the initial causes for ALS that wasn't really understood before that, and that would be low oxygen. So oxygen starvation to the brain. Also, there have been clusters of ALS, for example, in marathon runners or elite athletes of different types. There was a cluster of cases in Italian football players sometime back in the 90s, I think. So people have been looking for a connection between, intensive sporting activity and ALS for a long time, but no one's really found anything, because it's rare, because there aren't that many cases. But I think low oxygen could be one of them.
And what could be the cause of low oxygen to the brain?
Well, basically, the brain, think the brain is 10 or 15% of the body weight, but it uses 90% of the oxygen that you breathe in goes to the nervous system. So it's so energy demanding because every time a neuron fires, which is the way we think, or activity in the nerve system is through firing of neurons, you get a lot of pumps and things that need to be reset to reset the conditions in the neuron before it fired. I know it's a bit complicated, but if you think of a rocket, if you fire a rocket up into the sky, you need an awful lot of energy to restock it so it can fire again. You need to bring it back down. You don't do that with a neuron, but you need to refuel it. You need to set everything back, give it a nice polish and put it back so it can do it again. And this is happening every, microsecond in the brain in millions of neurons. So you have a huge demand for oxygen. So if there is low oxygen in the brain, then that can, the neuron itself is under stress because it's not able to make enough the oxygen leads to energy production, right? I didn't say that, but the mitochondria in the brain in the cells of the brain metabolize the oxygen to make energy. And that energy is important for all these pumps and downstream things. So you stop the oxygen to the brain for even a very short time, you get a lot of damage. And this is what happens in a stroke, for example, because the blood vessels are blocked, so you don't deliver oxygen to that region of the brain anymore. But what we found is that these actual low oxygen causes this protein that we were studying that can cause ALS if it's mutated to unfold and become very sticky, and then it sticks to these other friends of it, and then it creates this spreading protein spreading disease called a prion-like disease. And it's been discovered now that actually this kind of mechanism is happening in Parkinson's and in Alzheimer's. So it's first found in Creutzfeldt-Jakob disease, or the mad cow disease that you've heard about in the UK. This is a pure prion disease where the prion protein, as it's called, forms this new conformation, new shape. And when it does that, it gets its friends to do it. It's a very rare event, but once it happens, you can't really stop it. It goes spreads like a forest fire. And that's why Creutzfeldt-Jakob disease goes very quickly. And first symptoms, you can't find your way home for some reason, and then 6 months later, you're dead. So these similar mechanisms seem to happen in all neurodegenerative diseases, and that's why they creep along and get worse over time. So this is good. I mean, it's been a big discovery the last 5 to 10 years that this is happening in all neurodegenerative diseases, but still there's no way to really stop it yet. But there is hope. It just takes a long time, and there's very little research money really for neurodegeneration compared to cancer. It gets maybe 1/5 or 1 or 2% of the funding that goes to cancer.
Wow, that's weird, because I work with older people, and ALS and Parkinson's and Alzheimer's are pretty much the most common types of diseases, neurological diseases that we encounter, especially Alzheimer's. So that's weird that they don't found that more than they do.
No, I know it's I think it's a question. Cancer often affects younger people, so it's easier to motivate people to put research money into that, or charities. A lot of it comes in Sweden from Cancer Fonden, so there's a huge amount of charity. So a lot of people die of cancer compared to ALS or Alzheimer's, although those diseases are increasing. And therefore, a lot of people leave money to that kind of research. And also, it often affects young children. And that's a good way to, it's an advertising thing.
Yeah. I've read that you have, because I've heard, first time I heard about stem cells was on the Joe Rogan Experience for like 3 years ago, I think. And he spoke about it in a way when you get a injury or inflammation in the body. He talked about that you have the clinics down in like South America that you can go to, and then you can inject stem cells in that particular area. And then I don't know the exact process, but the area would heal and you will feel better. But I don't know if you had a critique against that.
Yeah, enormous one, actually, Charlie. But, well, had you asked me 10 years ago, I would have said just don't even go there, because, so what happened when the, stem cell field for humans started to boom in the early 2000s, because people could grow these things in a dish, right? It was very difficult to develop stem cells from humans in the way that had been done for mice. So I'll back up. In 1987-ish, 85, 87, the first mouse stem cells were made or isolated in a lab. And they used a couple of methods to do that, and it managed to be very successful. And what they could then do was kind of engineer those stem cells in a dish in culture. So they could, say, take away a gene or add a new gene and then put them back into a developing mouse embryo, and they would contribute to that developing embryo that would then be born. So you have something called a chimeric mouse then, which is half normal mouse and half from your engineered stem cells that you put in. And then if those stem cells contributed to the sperm or the egg of the male or female mice. You could breed them, and then the next generation would be pure engineered cells. So this was called knockout or knock-in mouse technology, or stem cell engineering. So it became a very useful method actually to study what would happen if you took out a gene in mice or in mammals, but could never get it to work in humans. It was very difficult because the same methods and the same culture systems for humans didn't work as they did for mice. But this was solved in early 2000s. And then in 2009, this paper came for these induced pluripotent stem cells that I mentioned. So here you could actually take a cell and turn it back into the early state stem cell. And at that point, the whole idea of, stem cell engineering for humans became very, very sexy. There was a lot of funding for it. Same as happened with COVID. It's a bit like kids playing football. When they're all running after the same ball, the funding goes here and everyone runs after the same ball. Yeah. So what happened was, because the regulation always takes time to catch up, countries that didn't really have any tight regulations about what you could put into a patient started these stem cell clinics, and they would then say, well, we can treat almost everything from migraines to MS to Parkinson's to diabetes. They would say, come in, we'll harvest your stem cells from your bone marrow or from your blood or from somewhere. We'll inject them into you and you will be cured of your disease. The problem is it almost never happened. Maybe it happened one in 1,000, placebo effect or just by chance. But many clinics offered these services, and I think patients were willing to take it, because if you've got an ALS diagnosis and you've got maybe 1 to 2 years to live and there's no hope, you will try anything. And these look very convincing. And it's called stem cell tourism, basically. I used to do a lecture about it, actually. So I think many people got burnt by this, but then sadly they've already died, so they're not going to do anything about it. They won't take the clinic to court, and probably the relatives are just grieving and they don't want to take the clinic to court. But what I would say now is the stem cell technology has moved on so much, especially in Japan, I think. So there are clinics there that will take your cells, turn them into stem cells, or isolate stem cells from, say, your bone marrow or from your fat tissue. Quite often that's a good place to get some stem cells from. So you just take subcutaneous fat and isolate stem cells that sit there. And then they can turn them into, for example, collagen producing cells and inject them into the bone. If you've got a bad knee joint, they'll inject them in there, and they do seem to graft, it's called, or integrate into the joint and do good things. So stem cells do have these amazing properties. If you put them in a diseased brain or a damaged tissue, it's a bit like if you have a party full of older people and you throw some kids in the room, everyone gets happier, everything seems to go well, because they release a lot of good factors that keep the surrounding cells kind of healthy. But it's not really well understood what those factors are yet. So there are good stem cell technologies now, I think, for like joint replacement and certain conditions, but not for like Alzheimer's, ALS, Parkinson's. I would say definitely not.
So stem cells are not a probable solution to ALS and Parkinson's and Alzheimer's in the future?
No, not yet. And I think there will be a big problem for those kind of diseases, because so what happens, let's say, with ALS is that the only cells that die really are these motor neuron cells. So they're the nerve cells in the brain and through the spinal cord that control the voluntary muscles, and also the breathing muscles, which are voluntary as well. And when they die, the connection to the muscle is lost, and then the muscle wastes away, because a little bit like when you have if you have a plaster cast on, if you break your leg, the muscles quickly wither away because you're not using them. So they have this, they need this constant contact with the nervous system in order to stay fit and healthy. So they disappear, but what's really only died are the motor neurons in the spinal cord. Now they grew when you were an embryo, when you were like a few millimeters long, they started to grow out and contact the forming muscles in the embryo. And when we're probably, 3 or 4 or 5 centimeters long, most of them are in place. So then we grow as a baby, the connection between the nerve cell in the spinal cord and the muscle gets longer, but the muscle, the neuron didn't change. It's the same neuron. So if that has then died and you need to replace it, you need to both put the neuron back in the spinal cord and get it to grow back to the muscle that it used to control, or that it now needs to control. In some people, that could be almost 2 meters if they're very tall, because you have some motor neurons that go, all the way to the muscles in the little toe. The scale of this is phenomenal. If you think the cell body, it's called, the main part of the motor neuron is Globen in Stockholm, the axon of some motor neurons will stretch all the way down to Rome. So that's the literal scale of the issue. Could you put something back in Stockholm and get it to grow all the way down to Rome? I mean, and you need to do this with thousands of these things. So very difficult engineering problem. So I don't think you could, once you get ALS, you've already lost about 90% of the nerve cells that control one particular muscle. So when you get symptoms, you can compensate until you lose 90%, and then you start to get failure. So someone might fall over, they trip up, they can't write properly. They've already lost a huge number. And this is one of the big problems also for other, Alzheimer's as well, that you've already lost 90% of the neurons before you see the problem. So the disease has been going on for decades, maybe for at least for years. And then you see the symptoms. And if you need to repair it, you need to put new cells back into an already damaged environment and improve it, but also get them to connect up to places where they connected when the scale of things was much, much smaller. So I think there's more chance then for kind of, I hate to say it because I don't think Neuralink is a very good technology, but something like Neuralink, where you could have a brain machine interface to recontrol the limb that's not working anymore through the brain. But the stem cell part won't really help for those diseases, no.
I read one thing when I did some research with ALS, and I read that mitochondrial stress could be a factor as well.
Yeah.
And one of the things that I then connected was that the mRNA vaccine could cause some mitochondrial stress. Is that true?
Yeah. Yeah, I mean, so generally, I would say, and that's one thing I've kind of understood much more in the last 5 years or so, and coming from our research, that what you see in neurodegenerative diseases is a metabolic shift in the brain or the cells that are affected before you start to see the symptoms. And even before you see any of these, molecular changes that I'm talking about, like protein clumping and, cells not behaving properly. So there's a metabolic change, meaning that nerve cells have this very high demand to make a lot of energy, ATP, so they can run all these pumps and things that I talked about. And that requires that their metabolism is working very well. They get all the raw materials in that they need. They can cycle them through to make all the things they need to repair. And if there's a problem in that manufacturing process, let's call it, then everything can grind to a halt. Or let's say the trash starts to build up down at the back of the shed and then there's a big pile of it. And before you know it, it's blocked the road. And once you've blocked the road, you can't get to Rome anymore. Then it's already a catastrophe. But the problem started because the man who was taking the bins out didn't get his breakfast. I think this is literally the way that, yeah. So in Alzheimer's, for example, one problem is that the nerve cells don't make enough ATP, enough energy through the mitochondria. And because the mitochondria are turning glucose into fructose, and the fructose actually inhibits the mitochondria themselves from making enough energy. So either you have too much fructose in your diet, and I don't mean eating too much fruit. It's kind of in the US, this high fructose corn syrup, a lot of lesk, a lot of fructose sugar drinks can lead to a metabolic state in the body where you're actually produce, or too much glucose, too high levels of glucose in the blood, diabetes basically. And then the neurons will turn that into fructose, and the fructose will stop them from making enough energy. So if you could reset the metabolism in that person, you probably could help at least stop the decline. You might not repair the brain because it's already damaged, but you might, reverse the problem.
So what you're saying is a healthy lifestyle can really cure a lot of diseases before they happen.
Yeah, I mean, we are what we eat, basically. That's one very important. What we put in ourselves, we become, because that's what we have as raw materials. And keeping, healthy, not being overweight, not smoking, not drinking too much, all these things in the end will help. A lot of it depends on our genetics and, how we deal with these kind of stresses that we meet in our lives. But diet is a huge thing, not just because of what you eat, but also because of your gut bacteria. So if you have inflammation in the gut, that can also then trigger inflammation in the brain. So a lot of people have gut inflammation. We eat a lot of wheat products, and wheat contains gluten, and gluten damages the lining of the gut. Even if we can tolerate it, it's still happening, which means we get a lot of slag product, a lot of wheat into the blood system. And then if you have an inflamed gut, you'll get inflamed brain. So there is a lot of evidence now that Parkinson's disease starts actually in the gut, and it spreads along the vagus nerve that goes into the back of the brain here, and then from there into the midbrain, where these dopaminergic neurons, which are the ones that kind of are very important for mood, but also for stabilizing the nervous system or our motor movements, that they die. And when we lose those, that's why Parkinson's people, they have this very hunched gait, often no expression on the face. They're like deadpan and tremor. Not all of them, but some. But that seems to start in the gut much earlier, maybe 20, 30 years before. And one way you can treat it, it seems now, is to do a fecal transplant, a poo transplant. So you take poo from a healthy person and you put it into the colon of a diseased person, and it reduces the inflammation because you're transferring the good bacteria, let's call them, from the healthy person to the diseased person. And there are clinical trials happening now, actually, in the EU, in Belgium, looking at this. They've tested it in mice. It works in a mouse model of Parkinson's. It reduces inflammation in the gut and then in the brain, and less of these cells die that would die in Parkinson's. So they're now trying it with Parkinson's patients to see if they can reduce the progression of the disease. And I think this could also work also for Alzheimer's and potentially for ALS, but maybe ALS goes very quickly. So once you've got it, it's difficult to correct. But Parkinson's is generally much slower. Anyway, so your original sorry, I went off on a bit of a ramble there.
No worries.
But basically, metabolic changes in Alzheimer's for sure, and in ALS too. So what you see is some of the mutations that trigger ALS push the cells into a different metabolic pathway regarding how much fat or sugar they use to generate energy. And depending on whether they're using fat or sugar to make energy depends on what things you can make with the byproducts of that process. So maybe motor neurons, need something from fat metabolism, let's say, that then is there protecting them from dying when they're under stress. If they're not using enough fat, they'll have less of that thing, and therefore they're more susceptible over time to dying and making these protein clumps, which then when that process starts, you can't really stop. So one thing we found in ALS patient cells compared to not ALS patient cells is what the earliest change we could see were these very long elongated mitochondria that just look crazy, actually. So there's some change in the mitochondria in ALS cells that happens before you see any other disease effects. And another thing that's very important is RNA in these motor neurons, because what happens in a motor neuron when it's under stress, it'll stop making new proteins. So it drags in all the RNA that's usually is the template for these proteins into these little granules called stress granules, and it keeps them there. stops making any protein till the stress has gone away, and then it releases them again. So it goes back to normal. It's a little bit, like there's a crisis at home, you pack all your bags, you wait for the crisis to go, and then you unpack everything again and off you go. So this happens in our nerve cells as well. So if you then introduce modified RNA in a lipid nanoparticle into a motor neuron, then that can really mess that process up. And I think that's why there have been a number of cases of very rapid onset ALS after the COVID-19 vaccination program. And I think this is one of the reasons. It's this RNA toxicity in ALS cells. Another effect could be on the mitochondria. So I know at least, not directly by 1 degree of separation. So I know the person that knows the person that has died of ALS, and at least 5 people that I know of now have all attributed their first symptoms to the week or the days after they got the first shot.
Has there been an increase in ALS since the first COVID vaccine ruled out?
There are reports from different countries. It's very hard to get the data, Charlie. This is what's most frustrating. I've written to clinicians in Sweden and asked them, and if you look on the database, there's something called neurodashboard on the neuroregister, which is the Swedish organization that collects all neurological diseases apart from Alzheimer's, because that's not seen as a neurological disease. It's a geriatric disease in Sweden. So Alzheimer's patients are seen in by geriatric doctors and not by neurologists. But in all other diseases, if you look at the number of diagnoses, you see a big spike in ALS in the last 3D, after 2001 and onwards. The only thing I'm not sure about, though, is if those numbers are absolutely accurate, because they seem a little bit lower than what you would expect for the total number of cases per year in Sweden. You'd expect like 200 new cases a year in Sweden based on the incidence of ALS. And the numbers reported into the neuro dashboard are less than that, but you do see a spike. So I don't know if that's just more people reporting from different hospitals in Sweden. I don't think so, because the coverage is very high. But when the people that really have this data are the ALS clinicians in Sweden, and I know other reporters have asked them if they've seen an increase, and they just haven't replied, or they say no, not really. But I know people that have definitely had ALS onset and died after getting the vaccine. Yes.
Yeah, you've been very vocal since the pandemic started as well. I know you've part of the Doctor's Appeal with Sven Roman. I interviewed him, and very interesting conversation. And also, I think you're part of North Group as well, and you've been a part of a lot of scientific articles. You've written about the RNA vaccine and its side effects. You wrote the article together with Sven Roman, which was connected the dots between the infertility rate we saw in 2021 and the COVID-19 vaccine as well.
Yeah. So I got it connected with Doctor's Appeal in 2022, end of 22, I think, Charlie, because I've been essentially, working on my own or being frustrated about these things I was seeing and scared, really, but there was no one to talk to, because no one in my work wanted to listen. I was branded as an anti-vaxxer and someone you should kind of steer clear of, because Jonathan doesn't think like the rest of us. And it was a pretty tough period, I have to say. But then late 22, I got in touch with Doctor's Appeal. And that's when this Sven and Katrine and others were organizing this conference in Stockholm in January 2023, which turned out to be a huge success. And then after that, I was involved in several other conferences. So there was another one in Stavanger, which was very infamous, because Elsa Widding was there and talked about climate politics and energy politics, really, not really climate. It was just like, very sensible, but because she's perceived as being a conspiracy theorist, people on the left or the center don't want to listen to her. They won't even comprehend what she said. But anyway, because she was on the stage with this English politician, Andrew Bridgen, and he'd made a tweet, retweeted something from a Jewish doctor, an Israeli doctor, that said, COVID-19 vaccines are worse than the Holocaust. Elsa was then branded as being anti-Semitic because she'd shared a stage with someone that shared a tweet. Is this school playground level of journalism that was, I've forgotten which, I won't call it news outlet, but which Swedish news outlet put this out. But so I got tied in with that because I was actually acting as chairperson on the stage for that conference.
So you're not only an anti-vaxxer, you're also anti-Semitic according to traditional
yes
media.
I must be, because I stood on a stage with somebody that retweeted something from an Israeli doctor.
I think I can't share a Zoom meeting with an anti-Semite and an anti-vaxxer, so I need to go now, Jonathan. No.
yeah shut me down.
Exactly.
So anyway, I was part of that thing, and then I started to work quite closely with Sven. I mean, we were looking at Sven had been tracking the number of births in Sweden per month before, so I'll back up slightly. One of my biggest worries when I first heard about mRNA vaccines using the spike protein was just how difficult it is to make any kind of, to turn on a protein of that size and that complexity in a cell, even in a dish, and do it very well is difficult, because it's a very big protein and it's 3 strands of the same thing wound together. It's called a trimer. So if I was going to try to make spike protein in my cells in the lab, in the dish, and I transfected them with some RNA, only a small proportion of what I made would be good, would be the right thing. There'd be small versions, there'd be unfolded versions, there'd be these clumpy versions, and the cell's going to produce these because it's trying to do things much faster than it normally would, and it trips over itself and makes mistakes. So I just thought this is a really bad idea, because people are going to be making many other proteins and the wrong proteins, as well as the spike protein that is designed. And every single drug company went after the same target. It was crazy. If you think of we want a broad strategy to come up with a new vaccine to stop this pandemic in its tracks, and everyone uses the same strategy. Even some people made it in DNA, some did it in RNA, some people put it in a virus, but it's still the same protein. So for me, that already smelt like there's something really wrong here. This is not logical. Anyway, then so I was worried, because the spike protein is very similar to a protein on the surface of our early embryo as it implants into the uterus, into the womb, called syncytin. And certain people had flagged up the point that these proteins are quite similar and they have a similar function. They get cells to fuse together. So maybe if you vaccinate people with spike protein, you're going to get antibodies that will block block implantation of the embryo. And okay, maybe the risk of this is very small, but you can't do that unless you actually have tested. You can't put this into a person, particularly a pregnant woman or a woman of childbearing age, if this is even a remote risk. And nobody had checked. So I was alarmed, and I wrote it already in like November, December 2020 to 5 or 6 people in the university saying, I'm really worried about this. What do you think? And one person replied, and they said, oh yeah, but I think they will have looked at that. The drug companies will have thought of that. No, they didn't. So Sven and I started looking at the data, and we noticed that the birth rate in Sweden was starting to drop, and it dropped exactly 8 to 9 months after the rollout of the vaccines to the fertile proportion of the Swedish population, and it's continued to drop. And you see the same pattern in many other countries. Sweden has not seen a big increase in excess deaths, though, like other countries, Germany, for example. But they have the same drop in birth rates. Now, this is something which has never been talked about on national TV, in the national press. There must be some kind of blanket order. You do not mention the birth rate in association with the vaccines, because you see these nice fluffy things on SVT where they've talked about, oh, Sweden's got a record low birth rate. We've never seen such a low birth rate since the 1800s. Isn't that terrible? What could it be due to? I think it's the Ukraine war. People just don't want to have children anymore. Well, if that was the case, they wouldn't be having sex. And if that was the case, you'd see a drop in abortions as well. But if you go to SCB and look at the data for abortions, there's been no change over the last 5 or 6 years. Same number happening like regular. Same number of miscarriages allegedly. And I don't know if that's true reporting or not, because I have heard stories about increased miscarriages, increased calcification of the womb, various problems in childbirth, but nobody's reporting those things if it is the case. So it's just rumors at the moment.
Yeah, because I've also heard, I don't want to interrupt, but the thing with the low birth rates right now in all of the Western world has been it's huge in media right now. They talk about it a lot.
Yeah.
And I've not really connected it to the vaccine before I talked to Sven, because I knew the vaccine had side effects according to some studies with your myocarditis and things like that, and sudden death, sudden heart stop and things like that. But I never connected it to the infertility rates. But my question is like, why? I understand why the US media won't report on negative side effects from vaccines and other drugs, because they have the commercials. It's a big thing in the US, like the pharmaceutical companies sponsor a lot of the media. But I don't get why the Swedish media doesn't report on these things. And maybe some journalists might connect it to the vaccine.
Well, I suspect, in the UK, they have this thing called a D-list, basically. So if the government goes out and says, you can't report on this, nobody reports on it. None of the established media, at least. And, really, they're the only ones that have a significant readership, or have been. I think people are changing now, because I don't read the newspaper or look at the TV anymore. I do it to see the rugby scores, and that's it. So I think things are changing, Charlie. But yeah, I think in Sweden there must have been a D-list thing gone out to the media to say you don't report on this. There's no way you're going to talk about it. And I haven't even tried to get data, because if I do, I know what I will need to apply for ethical approval, and what will come back is the response, well, no, we don't want to give you this because it'll affect people's willingness to vaccinate. Well, no shit. Yes, it will. People will refuse, but you should have thought about that before you roll that thing out into the global population. Can I show you some? I just want to show you an example, Charlie, of what's already happened. Right. So this isn't new. What happened during COVID? Can I share my screen? Is that okay?
Yeah, of course. Do I need to accept it, or? I think it's shared right now. I can see it. I hope the listeners can see it.
I'm just going to make this. So I made a few slides. I just wanted to explain this.
Of course.
So narcolepsy. I know you talked about this with Ann-Katrine and Boo when you interviewed them. But for people that don't know, this is a disease or a condition that affects your ability to sleep or stay awake, basically. You fall asleep at just like this. So you can be persistently sleepy, you can have disrupted sleep, or something called sleep paralysis, where you just basically, can't wake up and have hallucinations in that state. It's really not that well understood, but it can be related to this low levels of something called hypocretin, which is a sleep hormone that's produced in the brain when we go to sleep. It's incurable. So the treatment is really about managing symptoms and stopping the people that fall asleep having accidents when they would fall asleep just like this. So it's a lifelong problem, but as well as the falling asleep part, you have a higher risk of other health conditions like high blood pressure, depression, anxiety, ADHD. You're not sleeping properly, so the brain is stressed. But there was a huge spike in cases in Sweden in 2009 after the swine flu pandemic. And, this is something that Ann-Katrin talked about. So there was a report on this from Läkemedelsverket that came out in November 2020, right, just before the COVID vaccines came out on the market. And there's the link if anyone wants to go and read it, but I just summarized it over on the right. So in 2009, when the swine flu vaccine came out, pandemics, Swedes carved it up and did their best. So 5.3 million Swedish people took pandemics. I didn't, because I was allergic to eggs, and it was made in eggs, so I couldn't take the vaccine, fortunately. And none of the family did either, because we didn't really see the need. But 60% of the population did take it, because Swedes, do what Folkhälsomyndigheten tell us generally. I mean, we trust them, so we believe that they're giving us accurate information. So of the 31 million people in the EU that took it, 17% of all those doses were taken in Sweden. That just shows you, little Sweden, what an effect, or how much of the vaccine we took. So there was this report in 2013 from Smittskyddsinstitutet that the benefit of this the perceived benefit of this campaign was 100 deaths saved, 200 ICU cases. Well, you could say that's good. Out of 5.3 million, not that great. But the downside was that 394 cases of narcolepsy were reported into Läkemedelsverket as suspected side effects by 2020. So this is, 11 years later. In this report, though, and what Läkemedelsverket did was adjust this 394 down to between 150 and 200, because they said, well, these are just suspected and they've been reported, but we don't really, there is normally narcolepsy happening, even though it's very rare. Then we think it's about 150 or 200 cases. So, they've reduced the damage by 50%. This is what happens if you go into the neuro dashboard, and I think these numbers are accurate. So you see that red line, that's the spike in new cases of narcolepsy that happened in 2009 from the pandemics vaccination campaign. And it tails off down here into 2015. The graph on the right just shows you how complete this register data is. So for the debut datum, which is the new cases, it's about 80% coverage of all cases in Sweden. So it's not perfect, but it shows you what's happening. So I just put this into a table. What does this mean? What's the truth? Is it really 150 cases like Läkemedelsverket said, or is it 400? Sorry, the dogs are hungry, so that's why they bark. So what I did on the left, I put the year, I put the number of new cases in red are the ones after the vaccination campaign. I made the baseline, whatever it was in 2008. And then in the next column, you see the number of extra cases above the baseline. So in 2009, it was 98, 2010, 250, and so forth. And in the last column, you can see the total number of cases above what should have happened had there not been the pandemics vaccine. So you see 432 by 2014. So that's more like the 394 that was reported to Läkemedelsverket. It's higher, which you would expect, because not everyone reports all the cases to them. But they've somehow magic this down to 150 cases in 2020 when they write this report. So 11 years after the event. So, my conclusion to this is Läkemedelsverket, they're engaged in creative accounting. They're protecting the pharma industry. They are not protecting the Swedish public, which is what they're employed to do. And this is true for all medical regulators. European Medicines Agency, every Läkemedelsverket in every other country is doing the same thing. They're minimizing the damage that is caused by medical products in order to try and protect pharma, who incidentally pay most of their budget comes from the pharma industry in fees and things. So it's a systemic problem. It's regulatory capture at the highest order. And of course, little old Sweden, because we do what the government tells us to do, because we're good citizens. If they get captured and they're told to roll out this vaccine to all Swedes, then everyone's going to take it. And it'll be decades before the problem is really understood. So that was 2009, right? What happened in 2020 onwards is exactly the same, but on a much greater scale. And the diseases and conditions that these vaccines have caused, we don't know all of them yet, but we know there's a problem, and we see it in the fertility rate. We see it with myocarditis. We see it with arrhythmia. We see it in cancer now. It's starting to come. We're writing a paper on cancer incidence in Sweden. Certain cancers have started to turn up in 2021. But the establishment has just gone like this. We don't see anything.
Yeah.
And we're not going to discuss it either.
Yeah.
Which I find really distressing.
I'm quite an emotional person. I'm quite good at not acting on my emotions, but I get really emotional sometimes. And when all the time when I speak to people like you, to Ann-Katrin and Sven, when they talk about these statistics, I get really fucking angry. I get really angry because we have been lied to on such a high scale. So it's actually crazy. And Ann-Katrin told me about this narcolepsy, and she spoke to a person. I don't remember which person she spoke, but it's in the interview I did last week with her, last episode. And the person just said, and she showed these results on narcolepsy. And the person said, yeah, but it was a good test. It was a test for the pandemic to see how we could act in the next one. Yeah, 400 people. Was this in Sweden, right?
Yeah, well, this was Johan Carlson, that's general director of Folkhälsomyndigheten, that said this.
Yeah.
And I think this was a large part of the whole COVID vaccine rollout. It was run as a US military operation or a NATO operation, at least in certain countries. So in Germany, for example, there were military bases that were used to fly in the products, and then they were distributed out from military bases. And this was a logistics experiment. How quickly could we roll out a vaccine to the world population if we had to? So there are many aspects to this, which maybe we don't need to go into all of them here. But one of them was definitely that the elite, or the people in these positions, don't see us as human beings. They don't see those 400 mostly young children that had their lives. Well, Ann-Katrin said not destroyed. I would say destroyed. If you've got to live with a sleep disorder for the rest of your life is going to be shorter. You're going to have a huge burden of, things to put up with. And that was based on an experiment to see how quickly we could vaccinate Europe for a virus, which, if everyone had their, right levels of vitamin D and had a fairly healthy diet, would not have been that sick. It's awful, Charlie, but it's driven through fear, right? So public health has been weaponized now. It's been lifted up to the level of, we can control people by controlling, the response to perceived threats of a respiratory virus or whatever it is.
Yeah.
And those threads have all been connected up. And it came out of the US, sometime in the 90s, I think, or early 2000s, that they were going to start using pandemic health response, or what's it called? Security, health security is the name, right? You call it security, and then everyone should feel better that we're controlling. But what they're really doing is weaponizing normal diseases and pathogens against humanity by gain of function engineering them to make them more easily spread and more disease causing, and then releasing them at specific times to create chaos, which is what they did now.
Yeah, gain of function research originally in the 70s, where Richard Nixon put a stop to it, allegedly. It was bioweapon research. Kennedy has covered this a lot in his book, The Wuhan Cover-Up. They research on bioweapons, and the CIA continue with this after Nixon put a stop to it. And then, you have all these things with Anthony Fauci, and when Obama put a memorandum on gain of function in the US, he funneled it via EcoHealth to China. And then we had the Wuhan Institute of Coronaviruses, where maybe the first patient with COVID-19 came from. And then at the start, everyone who put forward this theory that it came from the Wuhan Institute of Virology of Coronaviruses or something like that it's called. They were conspiracy theorists, but now it's like pretty much
I know.
true that it came from this lab.
Yeah. Well, I mean, this is one of the things that made me realize that COVID was an engineered pandemic, let's say, because I looked very early on at the sequence of the SARS-CoV-2 virus. I made a lot of viruses in my career, working with them as research tools mainly. So I know how you cut and paste bits of them together to make them into the kind that you want to have. And I looked at the sequence very early on of the SARS-CoV-2 virus, and I saw telltale signs that it had been stitched together. And others did this as well, but they were, mostly on blogs that were, people didn't really follow or don't follow in a great deal. And then came this, dialectic, or these two sides. It's either released from a lab accidentally, or it came from the wet market. And we know the whole wet market hypothesis was a fabrication, because those emails between Anthony Fauci and Jeremy Farrar from the Wellcome Trust in the UK and others have been made available under FOE, where they're talking about how we're going to manage this narrative. So it's quite clear that this isn't, the real story. And, the fact that a pangolin and a bat suddenly got together in a wet market and gave birth to this virus that spread throughout the world is quite frankly ridiculous. Although, a lot of people have made careers out of it and got a lot of funding for selling this story.
Yeah.
It's propaganda.
And the real scary theory is if this was accidentally put out, or if it was
I don't think it was an accident. No, I think it was deliberately put out. And then you have the Omicron strain, which is totally unlike anything else that came before. Totally, but it's a completely new strain that originated in South Africa and then spread around the world really quickly, and essentially vaccinated everyone again, but with a real virus instead of an artificial construct. So, there are so many aspects to this that just don't fit with what I understand from my scientific background to be possible. So I think the chances that this was an accidental release is just, zero, or very close to zero, let's say.
And then,
but what did happen was a lot of cases arose after this Wuhan Games in October 2019, the Wuhan Military Games, right? Even in northern Sweden. So there is a case, there's a report in Norrbottens Kuriren and NSD from, I think, it came out in April 2021. I've got the article. It's still online. They haven't taken it down yet. But essentially, there were a bunch of people that had competed in the games in Boden. So they came back in October 2020 with very acute, strong respiratory symptoms. They were very sick. The doctors hadn't really seen this before, but then they got better because they were young, fit, and healthy, I guess. And then, the Smittskyddsläkare they were asking questions. Could this have been people infected in Wuhan already in October? Well, how did that happen if the first cases, started in December? It's impossible. There's also a researcher in Canada called Steve Pellesh, who's very good with antibodies and looking at antibodies binding protein. So he screened a lot of epitopes or small fragments across the SARS-CoV-2 virus against 4,000 blood samples of women from British Columbia, picked up in 2019, 2020. And he sees also the first reactivity against the SARS-CoV-2 virus happened around November. So there were already people getting infected, before we saw pictures on the telly of people falling dead in the street in China and, being covered up in the hazmat suits and swept away. So it definitely came out before the end of 2020. Yes.
Yeah, and it's a move towards totalitarianism, because I don't know if it was this year in May or last year in May that WHO, with Tedros as a captain, they discussed this pandemic treaty, which would label any atrocity or what do you call it, emergency.
Yeah.
That WHO label as a pandemic, and it can be like climate change or anything they label as a pandemic. They will then have total control over every country's response to it.
Yeah.
Fortunately, that time it didn't get passed, I think, but I think it's still on the table.
It's been passed in parts, and also the other part is this treaty, which really agrees to all countries sharing these pathogens. You know, rather than relying on EcoHealth Alliance to go and stick their hand up a bat's bottom and get a sample of virus and bring it back. Now, any country that owns these potential pathogens that could become pandemics, they can now share them quite freely. You know, so it's essentially bioweapon. The whole bioweapons treaty, what if ever we had one, is fallen apart because now everyone's at it. And, you know, it's quite easy to get grants for COVID to look at gain of function engineering, even in Sweden. You know, if you've got the right biosafety levels to work on it, you could easily do a project on that. I mean, what are we doing? Why? Well, I know what's happening. You know, science is now being co-opted also into this whole fakery of, you know, producing pathogens to create pandemics in order to lock people down. But we're losing complete track of reality. I can bet you that China isn't doing this. You know, China's working away, becoming the most technologically advanced, competitive country on earth, while the West is committing suicide by, you know, taxing carbon and, you know, wrapping us all up in these, putting us all, locking us down so no one can work anyway. But I would say this thing, Charlie. I mean, you just need to follow the numbers, right? The European Union, the EU were putting through their policy for a vaccination passport scheme. And I think that was due to be completed sometime in 2020 or 2021. And this was a 5-year program. So they already had a program to roll out a vaccination passport scheme. Sweden paid for all the vaccines. You know, we all paid on this contract that, for example, with Pfizer, that our head of the EU negotiated on WhatsApp with the head of Pfizer, secret contract, which tens of billions of euros for many billions of doses. So all those were paid for by the taxpayer, by the government. But what most people don't realize is that Swedish government paid 300 crowns per shot to the vaccinators, so the hospitals or the health agencies that did the vaccination, only if they put the people's details into the vaccine register. So we were paying through our tax money 300 crowns per shot in order to set up the vaccine register. Yeah, really, really dirty.
Yeah, it's crazy. I also wanted to bring up a study that you linked to me via mail, and it was the study in Czech Republic where they saw a correlation between infertility rates and the vaccine in women.
Yeah. So we wanted to carry on from what we'd seen in the nationally available public data about, you know, birth rates in Sweden to see, could we actually go back and find any data that showed that vaccinated women were less likely to give birth than unvaccinated women. The only place we could find that data was in the Czech Republic. Thomas Furst, who's one of the authors, managed to get that data from the Czech authorities through Freedom of Information. So we did this study together with Vibeke Manicke and Thomas Schmeling and Peter Rees Hansen. So these are Danish researchers that I've worked with on the side effects of the Pfizer vaccine in Sweden versus Denmark, where we saw basically 3 different clusters of, if you look at the reported side effects to läkemedelsverket and you look at the severity, so were they deaths, were they very severe or were they less severe, what you saw are 3 clusters of batches. So either you got a batch that was associated with a lot of deaths or very serious side effects or less serious side effects, and some were associated with almost none. So to me, what that says is that not all batches were the same, right? Because if they're all identical, then you would have the same spread of side effects reported for every batch. At least it's a signal that that might be the case, and that means that needs to be investigated. what Lekemedels-Verkit says is no, we can't. No, this is just no. There's no connection. You can't say that. Yeah, well, look into it then, but they won't. They'll just publishing meaningless papers about there aren't that many cases of myocarditis or on vaccination willingness. Like who are the people in Sweden least likely to become vaccinated? They're foreigners of a low educational level, basically. Me being one of them. Anyway, sorry, I'm digressing, but yeah, we first published a study looking at the side effects and could show that there was a huge amount of granularity in the type of batches that were injected and how many side effects they gave in Sweden, in Denmark, and the Czech Republic have shown the same thing. And there's been another study in the US as well. So there's 4 countries that all show the same. So then we looked at the number of births in the Czech Republic. So 1.8 million women, according to their vaccination status. So when they got vaccinated before, during pregnancy, did they have any births after that? And the answer is basically, I mean, you can't conclude that the vaccine reduces fertility, but what you can say, there's about 30% less births in women that are vaccinated versus the ones that were not.
And what's the reaction from the scientific community when you publish this study?
Well, it's twofold. I think the people that are let's call them awake. I don't like to use that term too much, but people that realize there is a problem are shouting about it, and everyone that is in denial, that claims that all vaccines are safe and effective, look at all the possible confounders and say, well, you never checked for ethnicity, for all these other potential co-founders that could explain the same thing. Why people that are sorry, it's my text. Why there are less births in the vaccinated than the unvaccinated. But what it shows, the data that Sven and I have published in Daily Skeptic, we didn't send it to a journal because we knew that nobody would publish it at that time, showed, say, a 10 to 15% reduction of births in Sweden overall. What this shows is that you only see the drop in the rate of births in the vaccinated. You don't see it in the unvaccinated. So either the women that are not vaccinated suddenly going, oh yeah, let's have more children because the world's much safer than it was in 2019, and we want to bring our children into the world, or they're actually just giving birth at a normal rate, and it's the vaccinated women that are not giving birth at the same rate. There are many studies that support that the vaccines damage fertility. There is an early study from peer-reviewed, published from Israel, showing that it decreased sperm motility in men by like 50%, I think, but significant reduction in sperm motility, which will affect fertilization. I've not seen anything to show that, what stage of pregnancy may be affected by the vaccines, but I suspect it's known that nanoparticles accumulate in the ovaries and the testes, and therefore one problem could just be kind of stress in the ovaries. It's been shown in rats that if you give the same vaccines to rats, you deplete their number of the cells in the ovaries that can produce eggs are depleted. So, potentially what we might see in the future is kind of exhaustion of eggs in younger women. So women going into menopause at a younger age. I mean, this is a catastrophe. If you want to cry, if you want to get angry, this is one thing you should get angry about, because this also could, be transgenerational. With thalidomide, it didn't really happen. So this was a drug given to pregnant women to prevent morning sickness. And it turned out that, it led to developmental defects in children, very short arms or limbs, and, deaths, of course. But the ones that were born, often these children survived and they were severely disabled. And that was very visible. And it didn't necessarily affect the next generation, but there is, I forgot the name of it, but there was another drug given in the 60s, 70s, which has been shown now that it not only affected the offspring, but the offspring of the offspring are also affected. It's a risk for breast cancer, I think. This could also be the case with the COVID vaccines. We don't know. We do not know what effect this might have on the next generation.
Yeah, no, it's dark, but I feel like it will be interesting to see the response from the public when the next pandemic comes, because it will come sometime in our lifetime. And I hope people are very much more awake now. And I hope we don't get brainwashed as we did this time. And I hope that the totalitarians and the elites, even though they are pushing towards this controlled society, I hope a lot of people already have woken up and are rebelling against it. That's the only hope we have, right?
Yeah, we just got to keep kicking back and pushing with all our, strength and humility and reason, to try to go to that world that many people think we live in, but we don't. I'm afraid we are totally lied to and manipulated at the moment in society, and that's unfortunate because this is probably the freest we've been in the last 70 years or so, and we're just giving it away.
Yeah. Well, Jonathan, is there anything more you want to cover, or?
No, I think that's good, Charlie. I think I need to go and
yeah, I know. But thank you very much for being here. Is there any place they can find you on media or somewhere that you want to plug?
No, I'm pretty anonymous, really, because well, it's a problem working in the university if I make myself too public on social media and say things that puts the university into disrepute. So I don't do that. I do have an X account, though. I think the best way to keep track of the things that we're doing through North Group, where we are trying to get governments we've written to the heads of state and health ministers for now 28 countries that have joined, asking them to put a moratorium on mRNA vaccines, to investigate why they were approved in the way that they were, and also to assure the populations that there's absolutely no risk of these damaging our DNA. So that's the best thing, and that's northgroup.info is where we put out the information about that.
Good. Okay. Thank you very much, Jonathan.
Thank you, Charlie.
Hope we can discuss this in the future as well. And
yes.
Thank you for listening, everybody. Bye.
Okay. Thank you.